So thank you for inviting me to give this talk today. I will talk about profiling of cells and tissues using highly multiplexed fluorescence imaging. And my background is within the Human Protein Atlas, where I've been working for many years to do just what this implies, profiling cells or proteins in cells. So I will talk a little bit about how we used to do it in the Protein Atlas, how we do it today, but more than move over to the highly multiplex methods that we are more and more working with today. And I should also say that I'm head of a spatial proteomics facility at SciLifelab in Stockholm, where we use these novel highly multiplex technologies as service projects to others. But to look back and talking about imaging the human proteome and what we started to do about 15 years of work, was the incentive that was described in the Human Protein Atlas, and this project was started by Professor Mattias Ullén. And the incentive of this was that we wanted to map all of the human proteins in cells and tissues and organs using antibody-based imaging, so that we can answer questions as of which proteins are expressed in what cell, where in the different cells are the proteins expressed, so at the subcellular level, but also on a more global level, like in what different tissue types. And to be able to do this, the whole project was divided basically in two parts, where the first part was about generating antibodies towards each of these protein coding genes, roughly 20,000 protein coding genes, and then applying immunohistochemistry and immunofluorescence for doing this systematic profiling of the human proteomes across 44 different tissue types and in a panel of different cell lines where the proteins were to be localized at the subcellular level. You probably all know this, so all of the data that has been generated within the Human Protein Atlas project is publicly available in the proteinatlas.org. So this is a database where all of the data is collected, and you can browse now more than 17,000 or images of more than 17,000 unique proteins from the human proteome. And besides all of the images, which are more than 10 million by now, there is also lots of RNA sequencing data, both from bulk samples, but also at single cell level. And the Protein Atlas, the content has really grown over the years. So now we have, I think it is as many as 6 sub-atlases where all of this information is collected into various sections to make it more user-friendly. Just to say a little bit about some of the conclusions that has been drawn from this massive project that has been going on for more than 15 years. If we start with the immunofluorescence that was used to map the proteins at the subcellular level, we have done this now for more than 12,000 or actually even more than 13,000 proteins. And we have looked at where these proteins locate inside the cells, and we are now able to distinguish between 30 different subcellular structures in various cell lines. And when we did this, about 50% of all of these proteins had never been studied before. So this was novel data that nobody knows before where the proteins were. And that also means that we can start to get more clues about what cellular processes these proteins are involved in. We know today from this work which proteins that are what we call organelle proteins, so which proteins are expressed in which of the 13 major organelles of the human cells. And one thing that is very interesting that we have found is that 50% of all of the proteins are multilocalizing, meaning that they are localized not only in one organelle but at several compartments, and that implies that they might have several functions, which makes the human proteome even more complex than what we had thought as for now. Another interesting finding from this is that we also see that 90% of all proteins show single-cell variability. So that means that we would have one staining pattern in one cell, but not in the cell next to it. And this is despite all of these cells are cultured in vitro and have the same DNA content and profile. And this is half of these proteins can be regulated or correlated to cell cycle variations, and that is why they express different profiles. But the 50% of those cannot be correlated to cell cycle, and this is, of course, a very interesting topic. It's something that my colleague Emma Lundberg and her lab is doing further research on. And this is just an example of such single-cell variability, where we can see a very clear on-off staining of this target protein, which is here represented in green, and the microtubules is here counterstained in red. If we move to the immunohistochemistry part and the more global mapping of the human proteome, this is a combined work of transcriptomics, but then the immunohistochemistry of over 17,000 proteins across 44 tissue types and based on around 24,000 different validated antibodies. And what we see here is that about 50% of all of these protein coding genes are localized or are expressed in all of the tissue types across the body. So they are what we call housekeeping proteins. And this also means that if we take a more like drug-centric perspective, 30% of all the protein targets that are now targets for drugs, they are expressed everywhere in the human body. So I think that this is information that can be used when trying to design novel drugs in the future, taking the aspect into in what part of the body is the certain protein expressed and what kind of condition is it that we really try to target with a new drug. If we look at the tissues that has the highest level of tissue-specific proteins, that is the genes that are expressed in testis, brain, and liver. And I said that this protein took about 15 years. This project took about 15 years to conduct until now. And that is because they're very, very massive amount of data. So if we look at immunohistochemistry, every single antibody towards a protein that we have mapped has been stained and generated 576 images. Because we do it in several samples for every tissue in question. And it's many validation rounds, etc. So it's a lot of work that we done if you consider that we can only look at one protein at a time. And these are just some representative images from one antibody that was in the project. And this is, I think, one of the main drawbacks for conventional immunohistochemistry, but also for many of the fluorescence methods that has been used historically, that it has a limited throughput in a way that we can only look at one protein, as in this conventional immunohistochemistry, or a few proteins at a time. And how many we can look at, I mean, it's slightly more with fluorescence, but still there is a limitation depending on the fluorescence, the spectral overlaps that we would get. So there is clearly a need for novel methods, and I will now move over to the highly multiplexed imaging and how this can be overcome. So just very quickly, I mean, why do we need a highly multiplexed imaging? And this is especially true if we look at tissue samples that are very complex. They consist of different cell types. The cells can have different cellular states. I mentioned the single cell heterogeneity and also how the microenvironment can influence the different cells. And if we look at tissues again, we might have also limited sample material, so we would like to maximize the information that we can get out from one single section. And today, for example, in clinic, in most cases, one look at the morphology just from the regular H&A stain or one protein at a time. But these novel kinds of methods could complement this information and get a better understanding, molecular understanding of what is going on at the single cell level. And there are several approaches for doing highly multiplexed imaging, and I would just touch upon two main tracks here. So the first one is the, let's say, continuation on the traditional immunohistochemistry, where one use chromogenic detection with thyroid chemistry. So we have a primary antibody, a secondary antibody that is conjugated to horseradish peroxidase, and then it is connected to a chromogenic dye. That is when you add a substrate translated to a dye, a chromogen that stays on the tissues, and that can be detected in a regular bright field microscope. And by using different chromogenic dye, one can do multiplexing, add a little bit, but it's still quite limited, as you can see in this picture here to the right, where they have four different markers shown in the very same staining. But again, this is not a higher plex than what we can also achieve with fluorescence microscopy. So the other track for doing this is to use somehow cyclic immunofluorescence protocol. So using fluorescence microscopy, but somehow cycle the process so that you will look at a few signals at a time and then do it in an iterative manner, and in that way overcoming the limitation in detection of different fluorophores. And two main ways of doing that would be either to conjugate the antibodies of interest with some kind of tag, usually a DNA tag, that can in turn be detected with a complementary oligo that is in itself labeled with reporter, for example, or fluorescent reporter. Or you can have the regular indirect immunofluorescence with secondary antibodies, and then these whole complexes would be added and imaged and eluted, as you can see on the image here to the left. And in my unit, the spatial proteomics unit at SciLifelab, we work with these different types of methods for highly multiplexed imaging, and this is our expertise area. And our mission as part of SciLifelab in Stockholm is to provide these novel technologies and the latest instrumentation for doing that, so that we can help researchers to do research that else would not be possible in Sweden, and we do it as a very subsidized price. I should say that we have some grants so that we could also do projects for European users. So if we start with one of the two methods that we work with in our lab, and that is cyclic fluorescence with Codex, and this is a platform from Ecoya Biosciences. I think it was definitely one of the first systems that were launched for automated highly multiplexed imaging. So we were beta testing this instrument together with Emma Lundberg's lab in 2018. And it has a microfluidics device that integrates with a microscope, and it uses oligotagged antibodies with the complementary reporters for detection in cycles. So you would have 3 proteins in every cycle, and I will show this on the next slide. And this method is compatible both for fresh frozen, paraffin embedded, or tissue microarray tissue samples. You could do it on cells as well. And using this technology, the inventors, the Gary Nolan lab, they have demonstrated as much as 40 proteins being analyzed in the very same sample. So this is what it looks like in a schematic way. So you would have different antibodies that each have a unique barcode conjugated to them. And then these barcodes have their complementary reporter that are labeled with 1 out of 3 different fluorophores. And these are the same fluorophores that you repeatedly use within your entire experiment. So you would have all of the antibodies. I think I'll show that on the next slide. Yes. You will have all of the antibodies that are being incubated on your tissue sections. You can see this panel down to the left, the Codex panel. So all of them are incubated at once. And then when the sample is put onto the microscope, the labeled complementary reporters will be added 3 at a time from a microtiter plate into this microfluidics chamber. So they will bind, and then the imaging will be done automatically. We usually run this overnight in our facility. And then after the cycles of all of the antibodies have been done, we get all of the images stacked, and we do stitching of them to put all of the tiles together. There is other image processing involving deconvolution, the flat field correction. And after that is done, one can do cell segmentation so that each and every cell in the tissue is identified, and then you will get quantitative data as intensity measurements for all of these protein targets that were included in your panel. So this is basically what this would allow you to do. So you would have all of your processed images, the cells are segmented, and this is done by the software also accompanying this Codex instrument. And once that is done and you have all of these intensity measurements, you can use those signals to define cutoffs and then define your different cell types using one or a combination of these different markers. And once you have done this, you can look at where all of these different cells are distributed in your tissue to look at cell interactions, for example, what cell types are close to each other and seem to interact. And then there is like a lot of analysis that one can do with this amount of data. And here is just an example of what the images themselves look like. So this is from a fresh frozen section of colon cancer core, where we stain this with 18 different antibodies using the Codex technology. And as I said, we could also do this on tissue microarrays. So this is the same. So whatever area is available within the imageable area of under the microscope, this is where we would get the data from. So as you can see here, we could not capture data from all of the cores, but we have around 25 cores in this same experiment that has been analyzed with all of these, in this case, 16 protein markers. The other method that I want to talk about, that is something that is completely new. So we will get this instrument in the beginning of next year and also start using that for service project. And this has the different approach. So here we use primary antibodies, where we can incubate 2 antibodies at a time if they are from different species. And then we would incubate with 2 secondary antibodies that are labeled with 2 different fluorophores. And then this complex will be imaged, and then the whole antibody complex will be eluted with a buffer of low pH and heat. And then for repeating that X number of times, we can build up a multiplexed image experiment. And all of this is done in this COMET instrument that is an integrated system that does both the staining and the imaging, so it's also automated. And I think one of the nice things with this is that there is no need for conjugation of primary antibodies, but as long as you would have antibodies that are working on this type of tissue, in your regular immunofluorescence, you could optimize them so that they would work in this platform as well, which is nice. So we have, for example, used antibodies from the Human Protein Atlas that we have generated in-house, and then started to build panels here to allow for multiplexed imaging. And even if this is a low threshold so that you can use primary antibodies, there is, of course, several optimizations that has to be done in order to combine all of these antibodies into one single experiment. And the first one is the sample pre-treatment. So all of the antibodies have to work under the same conditions, the antigen conditions, if we work on FFPE. So the pH, the temperature, and the time has to be optimized. And then after doing that, there is a titration of all of the antibodies that you want to include in the panel. So this is, of course, the concentration of the antibody, but we can also play with the incubation time of each of the markers. And after that, we also need to evaluate the elution time and the epitope stability. So if you remember the setup of this, we would have several cycles of adding antibodies and then eluting the antibodies with this low pH and heat. And we need to make sure that the epitopes that all our antibodies are to find and bind to are still intact after several rounds of elution. And when you optimize this, then you will eventually know which marker to start with at an early cycle and which markers that you can afford to take in the very last cycles because they are very stable. So here is just one example for when we optimize this. This is the fast cell surface death receptor in a colorectal cancer core. And this is about just to show the quality of the images that we get. So it's single cell resolution and like really nice. This is just one plain images, but they look really nice. And after optimizing all of these different antibodies and after evaluating to see that the staining is still the same after several rounds of elution, we can combine all of these stainings then and set up a program for staining them at the same time. It would look something like this, that we would have our markers, knowing which markers that we put together in one cycle and then in what cycle they are put. We have defined our working dilution for each of the antibodies and the corresponding concentration. And then also during the optimizations, also decided upon the exposure time upon the image acquisition. The elution time is the time for when where the buffer and heating is ongoing. And we can have a shorter elution time if needed, if the epitopes are more fragile. And here you can see also that like all of the proteins are detected either with Alexa 555 or 647, so only using 2 fluorescence channels. And this is just to show one example using 6 of the markers that we optimized on this colorectal core. So it looks something like this. It's a bit hard to appreciate all of the different colors at the same time, but this is just some glimpse of data that we generated on this system. With that, I'd like to end, and I want to thank MFM for inviting me to present here in this webinar series. And I also want to take the opportunity to acknowledge the rest of my team, everyone in Emma's lab and the Protein Atlas, and also all of our collaborators. I'm happy to answer any questions that you might have. Thank you.